Water breaks rules. Dozens of them. Not minor technical violations at the edges of chemistry. Fundamental, structural breaks in the behavior every other molecule follows without exception. Liquid water is so commonplace in everyday life that it is often regarded as a typical liquid. In reality, water is most atypical as a liquid, behaving like an entirely different material at low temperatures than when it is hot. You drink it, you swim in it, you watch it fall from the sky, and the entire time it is doing things that chemistry says it shouldn't be able to do. When you apply what we know about molecular structure to predict water's properties, the predictions are dramatically wrong. Water, and by implication life itself, should not even exist on our planet by the numbers. That's not a dramatic claim. That's the conclusion you reach when you actually run the calculations. A 3-atom molecule, 2 hydrogens and 1 oxygen, that breaks rule after rule, and every single one of those broken rules turns out to be exactly the thing that makes Earth livable. The discovery of the hydrogen bond can be dated to the early 1920s. This discovery opened the way to a new understanding of the chemistry and physics of materials. In the 1940s, Nobel laureate chemist Linus Pauling calculated that water, without the contribution of hydrogen bonds, should freeze at around -120 degrees Celsius. As strange as Pauling's conclusion may seem, it was one of the first scientific footsteps that uncovered a profound truth. Water hides many strange behaviors. -120. The liquid covering most of our planet's surface, the liquid filling every cell in your body, the liquid that falls as rain. By the cold arithmetic of chemistry, it should be a gas. Drifting in space, nowhere near Earth, nowhere near life. It sits on our kitchen counters in a glass instead of dispersing as vapor, because of a bond so strange that scientists argued about its nature for decades. A bond that bends the rules not once, but across the entire periodic table of properties. If you've ever wondered what the most studied molecule on Earth actually is, you're looking at it, or rather, drinking it. After centuries of observation, hundreds of thousands of published papers, and experiments filling entire floors of university libraries, scientists still cannot fully explain why water does what it does. Deciphering the precise molecular behavior of water across different temperature ranges is still a major challenge for the scientific community. That's worth sitting with. The most common liquid on a planet covered in liquid, and the molecular structure of it is still up for debate. Science has been circling this molecule for centuries and keeps finding new rooms in the same house. If that sounds like the kind of subject worth spending time on, subscribe, because every video on this channel goes exactly this deep, and water is just the beginning. Now, let's start with the molecule itself. Because everything begins here, with the shape of something so small, you could fit more water molecules in a single drop than there are stars in the observable universe. A water molecule is 3 atoms, 1 oxygen, 2 hydrogens. Oxygen sits in the middle. The 2 hydrogens bond to it, and based on simple symmetry, you might expect them to arrange symmetrically on either side, the way carbon dioxide works. Carbon in the middle, oxygens pointing away in both directions, charges canceling each other out. A clean, sensible geometry. Water doesn't do that. Contrary to a symmetric linear configuration, the molecular geometry of water is bent, V-shaped. This arrangement results from the oxygen atom's 2 lone pairs of electrons, which occupy space and push the hydrogen arms closer together, pinching the molecule into a crooked angle. The bond angle in a water molecule is approximately 104.5 degrees, notably less than the ideal tetrahedral angle of 109.5 degrees. 104.5 degrees, an awkward, asymmetrical, lopsided angle. And that specific crookedness changes everything. Why is water a polar molecule, while carbon dioxide with similar bond polarity is not? Geometry. Water is bent due to two lone pairs on oxygen, so its two bond dipoles don't point in opposite directions and can't cancel. Carbon dioxide is linear, so its two dipoles point in exactly opposite directions and cancel perfectly. Water ends up with a permanent charge imbalance, a negative side near the oxygen, a positive side near the hydrogens. Oxygen is more electronegative than hydrogen, attracting the shared electrons more strongly. This causes a partial negative charge near the oxygen atom and a partial positive charge near the hydrogen atoms. The molecule becomes a tiny permanent magnet, with a definite north and south always on. A molecule with permanent charges does something predictable. It reaches for its neighbors. The negative oxygen of one molecule reaches toward the positive hydrogen of another. They don't bond covalently. That would fuse them into a new compound entirely. Instead, they hover close, attracted but not merged, connected by something weaker than a full chemical bond, but far stronger than most forces between molecules. Hydrogen bonds are stronger than other intermolecular interactions, such as van der Waals forces or simple dipole-dipole interactions, requiring more energy to break. This is the hydrogen bond, the reason life exists on this planet.
Each liquid water molecule is involved in about 4 hydrogen bonds, with strengths considerably less than covalent bonds, but considerably greater than the natural thermal energy of the surrounding environment. 4 bonds. Not 1, not 2, 4, arranged in a geometry reaching out in all directions simultaneously. One water molecule connecting to 4 others at once, each of those connecting to 4 more, an interlocking web stretching in every direction through the liquid. What makes water behave anomalously is this particular tetrahedral arrangement, where a water molecule is hydrogen bonded to 4 molecules located on the vertices of a tetrahedron. A tetrahedron, a 4-pointed shape, like a 3-dimensional triangle, each point the location of a neighboring water molecule. Hold a ball and stretch 4 rubber bands out from it to 4 other balls arranged symmetrically around it. Each of those balls doing the same thing, and the next, until you've built a 3-dimensional scaffold filling every cubic centimeter of liquid. That's what's happening inside the glass of water on your desk right now. Not a passive soup of loose molecules drifting around, but a constantly reforming, constantly breaking, constantly rebuilding network of connections. Unlike in ice, these bonds are broken and reformed on average 1 trillion times per second in liquid water, so that the water molecules can be packed closer together and move very quickly. 1 trillion times per second. Every second you sit there, the water in the glass next to you is restructuring itself completely, over and over. It looks perfectly still. It is anything but. The first and most direct consequence of this molecular architecture, the one that led chemists to suspect something fundamentally different about water before they even understood hydrogen bonding, is the boiling point. Open a chemistry textbook and look at the pattern of hydrides, molecules made by combining hydrogen with elements from the same column of the periodic table. Group 16, the column containing oxygen. Work down from the heaviest. Hydrogen telluride boils at about -2 degrees Celsius. Move up to hydrogen selenide, boiling point around -42 degrees. Lighter still, hydrogen sulfide, -62 degrees. Follow the trend to its logical conclusion and predict where the smallest, lightest molecule in this series, water, should boil. The prediction? Somewhere around -80 degrees Celsius. A gas at room temperature. A gas at the temperature of a warm summer day. A gas at the temperature your blood currently is. The actual boiling point of water is 100 degrees Celsius, about 200 degrees higher than the trend predicts. So despite its small molecular weight, water has an astonishingly high boiling point.
Water requires more energy to break its hydrogen bonds before it can begin to boil. To push water from liquid to gas, you have to overcome all those billions of simultaneous hydrogen bonds fighting to keep molecules together. 200 degrees of deviation. Not a rounding error. A massive, life-determining departure from every trend chemistry would predict. If hydrogen bonding did not exist, water would be a gas at room temperature. No liquid water on Earth. No rain, no rivers, no oceans, no cells, no blood, no life. That single fact, delivered quietly in a chemistry lecture, almost never lands with the weight it deserves. The entire biological world, every organism that has ever lived on this planet, exists because water's boiling point is absurdly, anomalously, impossibly high. The only thing standing between a dead rock and a living world is a small electrical attraction between a slightly positive hydrogen and a slightly negative oxygen. A bond so weak it breaks 1 trillion times per second, but collectively strong enough to keep the oceans liquid. I'll be honest, the first time I genuinely understood this, I had to stop and sit with it for a while. It's not a dramatic discovery in a laboratory, it's a number in a table. -80 versus 100. A gap that is the difference between a dead rock and a living world. Now, you might think the boiling point anomaly is the most dramatic trick water has. It isn't. Not even close. The next anomaly, one that confused chemists for longer than the boiling point, involves what happens when you cool water down instead of heating it. Every substance you know of, when cooled, contracts. Molecules slow down, pack together more tightly, the material gets denser. The rule is universal. Cold things are denser, warm things are less dense. Water follows this rule perfectly normally, from room temperature down, contracting steadily, getting denser, behaving itself, until you hit 4 degrees above freezing. At 4 degrees Celsius, water reaches maximum density. The molecules are packed together as tightly as liquid water can manage. Then, instead of continuing to contract as temperature keeps falling toward zero, water starts to expand again. Cool it further, and it actually becomes less dense. By the time it reaches the freezing point, it is expanding instead of compressing. Why? Because those hydrogen bonds, bending and flexing and allowing molecules to slide past each other at higher temperatures, begin to lock into their preferred geometry as temperature drops. The bonds want to be straight, pointing in specific directions, and when they finally hold their preferred shape, the structure they form is more spacious than the one they occupied when constantly flexing and breaking. Order, paradoxically, takes up more room than disorder. This sounds like a minor physical curiosity. It is not. It is one of the most consequential physical facts in the history of life on Earth. Picture a lake in winter. The temperature drops, and surface water cools first, becoming denser, sinking to the bottom. Warmer water from below rises to replace it, gets cooled in turn, sinks again. The lake circulates until the surface water reaches 4 degrees, the temperature of maximum density. That 4-degree water sinks. After that, the surface water, now lighter than the water below, stays on top. It cools further, reaches zero, and freezes. Ice forms on the surface. The entire body of water beneath stays liquid, sitting quietly at around 4 degrees, undisturbed. Every fish, every insect larva, every microorganism living in that lake during winter survives because of this one quirk. The ice on top acts as a blanket, slowing heat loss from the water below. Since ice is less dense than the underlying water, it floats, forming a barrier and insulating layer between the warmer water and the colder air above. Now consider what happens if water behaved like everything else. If it continued to contract and get denser right down to the freezing point. If ice were denser than liquid water. Sinking ice would expose the underlying water to cold air temperatures, which would freeze that water in turn, which would sink, which would expose more water. A lake would freeze from the bottom up. Eventually, it would freeze solid. The winter sun isn't strong enough to melt ice sitting on the floor of a deep lake. It would accumulate, year after year, millennium after millennium. At scale, the same physics would apply to oceans. Polar oceans would freeze from the bottom. The ice would never thaw at depth. The oceans themselves would gradually fill with permanent ice.
Earth would look like Europa, one of Jupiter's moons, covered in miles of ice, inhospitable to complex life. The maximum density at 4 degrees above freezing. One quirk in one molecule's response to temperature. Quite possibly the single most important physical fact in the history of complex life on this planet. When water cools below 4 degrees and starts expanding, what happens when it actually freezes? Water's volume increases by up to 9% when it freezes under atmospheric pressure. Not a small number. Freezing water can exert forces strong enough to rupture a standard copper pipe, which is why frozen pipes don't just crack, they burst. But the same force that destroys plumbing in January is also, over geological timescales, one of the primary forces turning mountains into soil. Water seeps into cracks in rock. Winter comes. The water freezes, expands, wedges the crack open a fraction wider. Thaws. More water seeps in. Freezes again. After enough cycles, centuries, millennia, solid granite splits. The rock becomes rubble. The rubble becomes gravel. The gravel becomes soil. Without water's expansion on freezing, the weathering processes that create the topsoil life depends on would be vastly slower. At the molecular level, the reason for that 9% expansion is the same tetrahedral hydrogen bond network operating at a different scale. In liquid water, bonds can flex and distort without breaking, allowing molecules to sit relatively close together. In ice, those bonds lock into a rigid hexagonal lattice, forming cage-like structures with channels of near emptiness running through them. The structure is so open, so full of internal voids, that the solid form is less dense than the liquid. Like a skyscraper full of empty rooms being lighter than a solid block of the same material. Except in this case, it's the mathematical consequence of hydrogen bonds insisting on pointing in specific directions and refusing to compromise. This is why ice floats. Not just because it's a bit lighter, but because its entire crystalline structure is fundamentally more spacious than the liquid it came from. Although rare, certain other substances exhibit similar behavior. Silicon and bismuth also expand upon solidification. But neither of them covers 70% of a planet's surface. Neither of them fills every living cell. The anomaly exists elsewhere, but in water, it's not a curiosity at the edge of a lab experiment. It's the foundation of everything. Close your eyes for a moment and hold the picture of an alternate Earth. Same sun, same orbit, same rock beneath your feet. Water there behaves like wax. Dense solid, less dense liquid, solid sinking when it forms. The oceans fill from the bottom with permanent ice over the first billion years. Continental shelves become frozen strata that never thaw. The water cycle, evaporation, rain, rivers, sea, grinds to a halt. Life, if it ever started, has nowhere to go. The density paradox is not merely a chemical curiosity. It is a fundamental requirement for a habitable planet.
One molecule.
One bent angle of 104.5 degrees. One bond that forms and breaks 1 trillion times per second. From that, all the complexity surrounding you, every ocean, every river, every cell, every breath, becomes not just possible, but inevitable. And we haven't even touched what happens when water absorbs heat. There's an observation that sailors in the Mediterranean made long before anyone had a word for chemistry. Coastal cities, even in summer, stay cooler than the deserts inland. At night, the desert drops to near freezing while the shore stays mild. The sea holds its warmth long after the sun has set, long after the land has gone cold and dark. Ancient navigators noticed this pattern without being able to name it. They just knew. Stay near the water, and the temperature stays reasonable. What they were observing, without any framework to explain it, is one of water's most quietly consequential anomalies. Not as dramatic as ice floating or the boiling point contradiction,
But in terms of planetary impact, arguably more important than either of them. Specific heat capacity is the amount of energy needed to raise the temperature of a given amount of a substance by 1 degree. Water's specific heat capacity is among the highest of all known substances. It takes more than 4,000 joules of energy to raise the temperature of 1 kilogram of water by just 1 degree Celsius. Compare that to sand, which takes only about 800 joules, nearly 5 times less. Pour the same amount of heat into sand and water side by side, and the sand heats up almost 5 times faster. You've felt this. Step onto a beach in summer, and the sand scorches your feet within seconds of leaving the shoreline, while the water stays cool despite hours of direct sunlight. Drive inland from any coast, and watch the temperature on your car's thermometer climb steadily.
The sea doesn't just sit there looking blue.
It is actively stabilizing the climate of everything around it. Scale that up. The oceans cover about 71% of Earth's surface. They are an enormous heat battery, absorbing energy from the sun during the day and in summer, holding it, releasing it slowly at night and in winter. Oceans have a large heat capacity, which means they can absorb and store large amounts of thermal energy without undergoing large changes in temperature themselves. They act as heat reservoirs, distributing energy around the globe via ocean currents and moderating temperature swings in coastal areas. The Gulf Stream is the most dramatic illustration of this in action. Warm water from the tropics flows northeast across the Atlantic, carrying heat energy that originally entered the ocean near the equator thousands of miles away.
By the time that water reaches the coast of Western Europe, it releases its stored heat to the atmosphere, keeping cities like London and Bergen remarkably mild for their latitudes. London sits at roughly the same latitude as Calgary, Canada, a city that regularly hits -20 in winter. London rarely sees -10. The reason isn't the position of the land, it's the ocean current delivering liquid heat. Without the ocean's enormous heat capacity, the temperature difference between day and night on Earth would be vastly larger. The Moon gives a direct comparison. Sitting at essentially the same distance from the Sun as Earth, receiving almost identical solar radiation per square meter, but with no water and no heat buffer, the Moon sees daytime equatorial temperatures around 127 degrees Celsius, and nighttime temperatures near -173 degrees. 300 degrees of variation. Earth's average surface temperature variation is measured in tens of degrees. That difference is largely water. Now push the thought further. Suppose water didn't have this anomalously high heat capacity, that it absorbed and released heat at the rate of sand or rock. The oceans would heat up rapidly under summer sun, releasing energy back to the atmosphere in waves. The poles would freeze more deeply in winter. The atmosphere would swing between extremes, sweltering summers, brutal winters, with little of the temperate middle ground that complex land-based life requires.
The comparison to Venus is not hyperbole. Venus and Earth are nearly the same size, formed from similar material, orbit at relatively comparable distances from the same star. Venus does have water, or rather, it once did. Models of Venusian history suggest the planet may have had liquid water in its early history, perhaps for several 100 million years before conditions changed.
Without liquid water to absorb and redistribute heat, without the carbon cycle that requires liquid water to draw carbon dioxide out of the atmosphere and lock it into rock, Venus spiraled into its current state. Surface temperature on Venus today? Around 465 degrees Celsius. Hot enough to melt lead. Atmospheric pressure 90 times that of Earth. Raining sulfuric acid. The difference between Earth and Venus is not just position or size. It is, to a significant degree, water's ability to hold heat. What makes water's specific heat so high goes back to those same hydrogen bonds. When you heat water, much of the energy you add goes not into making the molecules move faster, which is what temperature actually measures, but into breaking and reforming hydrogen bonds. The energy disappears into the bond-breaking process rather than raising the temperature. Water requires more input to heat up because it has more internal structure to disrupt. When it cools, all that energy has to come back out. It releases heat slowly and steadily. A lake in autumn releases warmth to the air for weeks after summer ends, long after the sun has lost its intensity. The same molecular stubbornness responsible for the boiling point anomaly is also responsible for stabilizing Earth's entire climate. One molecular feature, two planetary consequences, and dozens more still to come. That molecular stubbornness has another expression you can demonstrate on a kitchen counter in about 30 seconds. Take a needle, a thin sewing needle. Lay it on a piece of paper towel and gently lower it onto the surface of a glass of water. Let the paper towel saturate and slowly sink. If you've done it carefully, the needle stays on the surface. It doesn't float. It's denser than water and should sink by every rule of buoyancy. But it sits there, resting on an invisible skin, a slight depression visible around it if you look at eye level.
What's holding it up is surface tension.
The same force supporting a steel needle is the same force carrying water from the roots of a redwood tree to its highest leaves, more than 100 meters above the ground, without any pump at all. Surface tension arises because water molecules at the surface don't have neighbors above them. In the bulk of the liquid, each molecule is surrounded on all sides by other molecules, forming hydrogen bonds in every direction, up, down, sideways. The pull is equal in all directions, and the molecule sits in equilibrium. But molecules at the surface have only air above them. The bonds they form are sideways and downward only. That asymmetry creates a net inward pull, a tendency to minimize the exposed surface area by pulling surface molecules tighter together. The surface behaves as if a thin elastic membrane is stretched across it. Water has one of the highest surface tension values of any liquid, exceeded only by liquid mercury among common substances.
A water strider doesn't just touch the surface lightly.
It stands on this molecular membrane, its weight distributed across 6 legs, each dimpling the surface without breaking it, essentially walking on the structural skin of water. Surface tension alone doesn't explain how water climbs. For that, you need one more property working in combination, and together they seem almost designed for the purpose of moving water upward. Capillary action is water's ability to flow upward through narrow tubes or porous materials against gravity without any external force. It depends on 2 things: cohesion
water molecules sticking to each other via hydrogen bonds, and adhesion, water molecules sticking to the walls of whatever channel they're traveling through. If the material is one that water molecules can form attractive interactions with, like glass or cellulose, the water molecules at the edge of the tube are pulled toward the walls, dragging the water column up behind them.
The narrower the tube, the stronger the effect relative to the weight being lifted. A tree has no heart, no pump. What it has is an elaborate system of tubes, xylem vessels, extraordinarily narrow, often just tens of micrometers in diameter. At those diameters, capillary action is powerful enough to pull water from the soil into the roots and up into the trunk. In a coastal redwood, water has to travel over 100 meters upward. Capillary action alone can't do all of that at that height. Water tension in the xylem column and evaporation from leaves pulling the column upward also play essential roles. But the foundation is always the hydrogen bond network, the same molecular attraction between water molecules and the surfaces they touch. Cut down a tree in winter and look at the wood. Those tiny rings of xylem vessels are the architecture that water carved out for itself over millions of years of evolution. Every fiber designed around one molecule's tendency to cling to itself and to anything nearby. This tendency also explains why a paper towel absorbs a spill, why a sponge works, why soil retains water, why seeds can draw moisture from the ground. Capillary action isn't special to trees. It's everywhere water meets a narrow space, pulling itself upward and into corners that physics says it shouldn't reach. These same forces, cohesion, adhesion, hydrogen bonding, make water the most powerful solvent on Earth. Universal solvent. Most people file that away as a piece of chemistry trivia. They shouldn't. Water's ability to dissolve things is one of the most important properties in the known universe. And the reason it works goes back to that bent angle, those partial charges, and those insistent hydrogen bonds. Table salt, sodium chloride, is a crystal. At the atomic level, it's a rigid, organized grid. Sodium ions with a positive charge, chloride ions with a negative charge, alternating in all three dimensions, locked together by electrostatic attraction. To dissolve it, you need something that can compete with the electrical forces holding the crystal together. Water molecules have partial charges. When a water molecule approaches the surface of a salt crystal, the negative end, the oxygen, is attracted to the positive sodium ions. The positive end, the hydrogens, is attracted to the negative chloride ions. Water molecules wedge themselves between ions, tugging in multiple directions simultaneously. As more arrive, each one pulling at the crystal's surface, the ions at the edge are eventually torn free and surrounded, wrapped in a shell of water molecules called a hydration shell. This is what dissolving actually looks like at the molecular level. Not a disappearing act, but a dismantling. Ion by ion, each one immediately caged by water molecules holding it in solution. A non-polar liquid, like oil, has no partial charges, makes no electrical offer to the ions, cannot compete with the forces holding the crystal together. Oil doesn't dissolve salt. Water does.
Consider what this means inside a living cell. Every biochemical reaction in your body happens in aqueous solution. The ions that carry electrical signals through your nervous system, sodium, potassium, calcium, exist in the fluid inside and outside your cells because water dissolved them and keeps them dissolved. The proteins that do the actual work of biology fold into their three-dimensional shapes partly because of how water interacts with their surfaces. The sugars powering your muscles are dissolved in your blood because water's solvent power keeps them in solution. The waste products of metabolism are carried away and concentrated in urine because water dissolves them. Without water as a solvent, the chemistry of life cannot run. Not in a different, slower, less efficient way. It cannot run at all. Water's dissolving power also extends to gases.
Oxygen and carbon dioxide dissolve in water, allowing your blood to carry oxygen to your cells and transport carbon dioxide away. Without water's capacity to hold dissolved gases, lungs and gills would be useless structures. Over geological time, this same dissolving power dismantles mountains. Rainwater dissolves carbon dioxide from the atmosphere as it falls, becoming slightly acidic. When this weakly acidic water contacts rock, it begins dissolving minerals, slowly, relentlessly, molecule by molecule. Limestone caves are carved entirely by slightly acidic water pulling calcium carbonate ions from rock over millions of years, leaving hollow chambers behind. The grand cycling of elements through Earth's geology, calcium, silica, phosphorus, iron, is driven fundamentally by water's willingness to dissolve almost anything it touches. Now, we've been circling around one more anomaly that ties these properties together, something that operates invisibly, but whose consequences you've felt every time you've been hot. Return to the boiling point, 100 degrees Celsius, the absurdly high boiling point driven by hydrogen bonds. But the story of what happens at the boundary between liquid and gas is stranger than that number alone suggests. Even below 100 degrees, individual water molecules at the surface constantly escape from the liquid and become vapor. This is evaporation, not boiling, which happens throughout the liquid, but the quieter process of surface molecules gaining enough energy to break their hydrogen bonds and fly free. What makes this remarkable is how much energy each escaping molecule carries away. The heat of vaporization of water is extraordinarily high, much higher than most common liquids because of the strong hydrogen bonding that must be overcome for molecules to escape. When a molecule of water evaporates from your skin, it takes that energy with it, stolen from your body. Every drop of sweat that evaporates is a measurable cooling event. A small amount of evaporation removes a large amount of heat from the skin surface because each evaporating molecule carries away an exceptional amount of energy. Sweating only works as a cooling system because of water's anomalously high heat of vaporization. A liquid that evaporated easily at lower energy cost would cool the body far less efficiently. You'd have to produce enormous volumes of liquid just to lose a small amount of heat. Human endurance physiology, the ability to cool the body during sustained high-intensity activity, is built on this one molecular property. The Hadza of Tanzania, one of the last hunter-gatherer communities on Earth, still practice persistence hunting, tracking large animals through the heat of midday until the animal collapses from heat exhaustion. The human, sweating constantly and cooling via evaporation, outpaces the animal that cannot shed heat efficiently. A survival behavior practiced for hundreds of thousands of years depends directly on water's stubborn reluctance to become a gas. By the early 20th century, all of these anomalies had been individually documented. Chemists knew about the boiling point, the density maximum, surface tension, the specific heat, the solvent power. They tended to treat each anomaly as a separate curiosity, an interesting quirk of this particular molecule, worth explaining but not necessarily connected to any larger pattern. Then, in 1913, a Harvard biochemist named Lawrence Henderson published a book called The Fitness of the Environment. His argument was not simply that water had convenient properties. It was that these properties, taken together, formed an interlocking set of preconditions. That life as we know it is possible specifically because water departs from the rules in precisely the ways that organisms require. Henderson was a physiologist and biochemist who had spent years studying the chemistry of blood and tissue fluids. He wasn't a physicist trying to explain molecular structure. He was a biologist looking at the requirements of living systems and asking, what would you need if you were designing a solvent for life?
You'd need something that stays liquid over a wide temperature range, something that moderates temperature rather than amplifying extremes, something that expands when it freezes so that ice stays on top, something that dissolves the materials biochemistry requires, something that moves through narrow channels against gravity, something that removes heat efficiently when organisms overheat. Water satisfies every item on that list. Every single one. Henderson's contribution was not just to enumerate these properties, but to argue that their co-occurrence in a single molecule was not coincidence, but precondition.
He wrote that the properties of this cosmic substance are of a character singularly fitted to make it a source of life. The fitness of the environment was his argument. The environment, particularly water, is fit for life in a way that goes beyond what random chemistry would be expected to produce. The scientific community's reaction was complicated. The problem wasn't that Henderson's chemistry was wrong. His observations were accurate, and his cataloging careful and thorough. The problem was interpretive. His argument carried a faint scent of teleology, of purposefulness in nature, and that made mainstream science uncomfortable. Suggesting that water was fitted for life sounded, to some ears, like an argument from design. For much of the 20th century, that association kept Henderson's thesis at the margins of mainstream biochemistry. His observations were used as data. His broader argument was quietly set aside. It wasn't until the late 20th and early 21st centuries, as researchers in astrobiology began systematically investigating what conditions life requires to arise elsewhere in the universe, that Henderson's core insight was rehabilitated. Astrobiologists generating the list of requirements for a life-supporting solvent found it matched water's anomalies point by point. The framing shifted. Water doesn't just happen to work for life. It works in a specific, non-obvious, highly unusual way that most molecules don't. And the precise set of anomalies it exhibits are the same set that life requires. When scanning other planets and moons for signs of life, the presence of liquid water remains the primary target. Not because of tradition or habit, but because no other known molecule satisfies as many requirements simultaneously. Water isn't just the solvent life uses. It may be the only molecule in the universe that could play this role.
Henderson saw the pattern before the tools existed to explain why the pattern was there.
He catalogued water's fitness in 1913 and waited for a scientific framework that wouldn't arrive for another 100 years. And even now, with all the tools available, quantum chemistry, molecular dynamics simulations, ultra-fast spectroscopy, there is something about water that still doesn't quite fit. Something about how water molecules arrange themselves in liquid form, how the network structure changes with temperature. What exactly happens in that 4-degree window near freezing? These remain areas of active, unresolved research. The deeper you look into water, the more it resists a clean explanation. Right now, somewhere in a laboratory at the University of Washington in Seattle, a spectrometer is watching water. Not a glass of it, not a river of it, a layer just a few molecules thick, sitting against the surface of a gel membrane. The instrument fires pulses of infrared light at this layer and measures what comes back. What comes back doesn't match what the equations predict. The water near the surface is doing something the textbooks say it shouldn't be doing. It has been doing it for decades. For most of those decades, almost nobody wanted to hear about it. The scientist who first published on this anomaly in a systematic way was not looking for a new phase of water. He was looking at mussels. Gerald Pollack arrived at the University of Washington as a biomedical engineer in the 1970s, focused on understanding how muscle fibers contract. Muscle contraction was, by the late 20th century, considered essentially solved. The sliding filament theory explained how protein fibers in muscle cells pull against each other to generate force. Neat, mechanical, well documented.
Pollock accepted this framework the way most researchers do, as settled ground beneath his feet.
But something in the data kept bothering him. The water inside muscle cells, near protein surfaces, wasn't behaving like bulk water. Its properties were subtly different. Different viscosity, different electrical behavior, different response to heat. The standard model said this was a minor surface effect, a thin molecular layer of slightly perturbed water that quickly blended into normal bulk liquid a few nanometers out. Pollock kept measuring. The perturbed zone was far larger than anyone had predicted. Not a few nanometers, hundreds of micrometers in some cases. Visible under certain conditions to the naked eye. He spent years checking his equipment before publishing, because what he was seeing implied something that would be deeply unwelcome to the biochemistry establishment. That water near biological surfaces might be organized into a distinct phase. Not liquid, not solid, not vapor, but something in between. He called it exclusion zone water because it excludes solutes, pushing dissolved particles away from the surface into the bulk water beyond. Water in this state was more viscous than ordinary liquid water, had a different absorption spectrum for light, and carried a net negative charge. The reaction from the scientific community when Pollock's book appeared in 2013 was not enthusiastic. The criticisms were direct. Extraordinary claims require extraordinary evidence, and the idea of a previously undetected phase of water surviving a century of intensive study seemed implausible on its face. Some dismissed the work outright. Others were more measured, but deeply skeptical. Pollock's response was that evidence for anomalous near-surface water had shown up repeatedly in the literature, in strange results that researchers had noted and then filed away as experimental artifacts or surface effects.
The history of water science is partly a history of anomalous results reclassified as errors rather than signals. He documented dozens of cases where water near surfaces behaved in ways that standard theory couldn't account for, and argued that the standard response, assumed the equipment was wrong, had obscured a real phenomenon for generations. The scientific status of EZ water is still contested. Pollock's work has been replicated by some groups and disputed by others. The debate is ongoing. But what makes the story worth telling isn't whether Pollock is ultimately right about every detail. It's what the controversy reveals about water itself. Researchers are arguing seriously, in peer-reviewed journals, at international conferences, with real experimental data, about whether the most studied molecule in chemistry has an entire phase that was missed. After everything we know about water, after centuries of measurement, the argument isn't over. The deeper question underneath Pollock's work reaches far beyond his laboratory. If water organizes itself differently near biological surfaces, near proteins, near cell membranes, near the structures life is built from, then biology has been misunderstanding its own solvent for over a century. The water inside a living cell might not be a passive background liquid in which chemistry happens to occur. It might be an active participant, structured by its interactions with biological molecules in ways that influence how those molecules behave. This idea has a long history. The Nobel laureate Albert Szent-Györgyi, who discovered vitamin C and contributed foundational work to our understanding of cellular respiration, spent the last decades of his career arguing that intracellular water was fundamentally different from bulk water, and that this difference was essential to how life worked. He was largely ignored by the mainstream biochemistry of his time. The tools to test his intuition didn't yet exist. Now they do. Researchers using those tools are finding, cautiously and with significant uncertainty, that something about intracellular water remains unexplained. Which brings us to the question haunting all of this.
Not how water sustains life as it exists now, but how life began. 4 billion years ago, there was no biology on Earth. No cells, no proteins, no DNA. The planet had liquid water, oceans of it, possibly from very early in its history. In that water, something happened. Molecules collided. Some stuck together in ways that allowed them to replicate. Information, in the chemical sense, began to store and copy itself. Life, by any reasonable definition, started. The central mystery of the origin of life is not just what molecules were involved. It's how the transition happened, how chemistry became biology, how non-living matter organized itself into something that could maintain itself, reproduce, and evolve.
One thing that emerges clearly from origin of life research is that water isn't just the backdrop of this story.
It's an active part of the plot.
Researchers studying the origin of life have tried conducting relevant chemical reactions in non-aqueous solvents, in formamide, in ionic liquids, in other liquids that could theoretically dissolve organic molecules. Some reactions work in these alternatives. Many don't. The specific chemistry of early life seems tuned to water's particular combination of properties in a way that's difficult to replicate. Not just the solvent power, but the hydrogen bonding, the specific heat, the way water molecules interact with organic surfaces. Take RNA, the molecule that many origin of life researchers believe was the first self-replicating and catalytically active molecule, predating DNA and proteins. RNA folds into complex three-dimensional structures that give it catalytic function. These folds depend on hydrogen bonds between different parts of the molecule and on the interaction between the molecule's surface and surrounding water.
Remove the water, substitute a different solvent, and the folds collapse or fail to form. The molecule becomes chemically inert. Water doesn't just dissolve RNA. Water participates in making RNA functional. There's something tragic about imagining this chemistry tried in the wrong solvent. The molecules present, the energy sources available, even the right concentrations, and nothing happening. The dance requires a very specific floor. The origin of life may not just have needed a solvent, it may have needed exactly this one. If you built a parallel planet, same organic chemistry, same raw materials, but filled the oceans with some other liquid, even a liquid with decent solvent properties, the precise sequence of events leading from simple molecules to self-replicating systems might never have occurred. Not because the chemistry was impossible in principle, but because the choreography required a very specific stage. Water wasn't just present at the origin of life. It may have been the director. The possibility that life requires water, not just a solvent, but specifically water, changes how we search for life elsewhere in the universe. Picture the view from the Galileo spacecraft in the late 1990s, making its closest passes over Europa, the fourth largest moon of Jupiter. What the instruments saw was ice. A global surface of fractured ice, crisscrossed with reddish-brown lines, smooth in places, chaotic in others, with almost no impact craters, meaning the surface was young, constantly being renewed by something happening below. Tidal heating. Jupiter's enormous gravity pulls on Europa with changing force as the moon orbits, flexing and stressing the interior. That mechanical energy becomes heat. Models suggested this heat could be maintaining a liquid water ocean beneath the ice shell, potentially more than 100 kilometers deep, containing more liquid water than all of Earth's oceans combined, kept liquid not by sunlight, but by the grinding of gravity. The scientist who made the case most publicly for a liquid ocean beneath Europa's ice was a geophysicist at the Jet Propulsion Laboratory, who had spent years studying extreme environments on Earth
where life persisted in conditions far outside what anyone had previously thought possible. He had been to Antarctica, to the Atacama Desert, to the deep-sea hydrothermal vents discovered in the 1970s, places where life existed without sunlight, without moderate temperatures, in conditions once considered definitively sterile. Those discoveries had taught him a lesson that transformed astrobiology. Don't use Earth's comfortable surface as your template for life's requirements. The requirement is not warmth or light or moderate chemistry. The requirement is liquid water. His argument about Europa was simple and pointed. If there is liquid water, there is the possibility of life. Not the certainty, not even the likelihood, the possibility, which is more than we can say for almost anywhere else in the solar system. Since then, the case for a European ocean has moved from speculation to near certainty. Hubble Space Telescope observations detected water vapor plumes erupting near Europa's south pole, suggesting material from the ocean below occasionally vents towards space. These plumes matter enormously. They mean water from a potentially habitable ocean may be accessible without landing or drilling. A spacecraft flying through the plume could sample it directly. The Europa Clipper mission, launched in 2024, is currently traveling to Jupiter. Among its instruments are mass spectrometers capable of analyzing the chemical composition of any plume material it encounters. If the ocean below Europa's ice contains the organic molecules and chemical gradients that life requires, the mission may detect signatures of that chemistry. Not proof of life, but evidence that the conditions are right. The density anomaly keeping fish alive in winter lakes, the same physics operating at planetary scale, keeping a possible ocean liquid beneath the ice of a moon 500 million kilometers from the nearest sunlight. One molecule's quirk, playing out across the entire solar system. Then there's Enceladus, Saturn's small, bright moon, which in 2005 became the most exciting object in the outer solar system when the Cassini spacecraft discovered active geysers at its south pole. Water ice and vapor shooting hundreds of kilometers into space, carrying organic compounds, molecular hydrogen, and silica particles, the chemical signature of water reacting with rock at high temperatures. Hydrothermal vents. On a moon of Saturn, liquid water in contact with hot rock, producing chemical gradients. The same environment many researchers believe was the cradle of life on early Earth. The salt and pressure physics of that ocean are different from Earth's oceans, in ways that matter. Enceladus's ocean sits under an ice shell tens of kilometers thick. Under that pressure, the freezing point of water drops. Water remains liquid at temperatures below 0 Celsius, kept from freezing by the weight of miles of ice and ocean above it. Salt lowers the freezing point further. These are the same physical properties, water's anomalous response to pressure and dissolved solutes, that make oceans remain liquid in environments that would seem impossibly hostile by any surface world standard. The map of potentially habitable environments in our solar system has redrawn itself entirely in the last 30 years. Every redrawing was made possible by water's anomalous behavior. The ice-covered ocean moons of the outer solar system exist as candidates for life specifically because water refuses to freeze solid under pressure, because ice floats rather than sinking, because water's specific heat allows geothermal energy to be distributed and retained rather than immediately lost. Back on Earth, in laboratories where instruments are now precise enough to watch individual molecules move, something quietly troubling is emerging. The more carefully researchers look at water, the less certain the picture becomes. For most of the 20th century, the structure of liquid water was described by a single continuous model, a hydrogen-bonded network, constantly breaking and reforming, with a statistical distribution of bond angles and lengths. Disordered, fluctuating, but averaging out to an arrangement that broadly explained water's bulk properties. Most textbooks still present this picture. The trouble started in earnest in the 1990s and accelerated in the 2000s,
as X-ray spectroscopy became precise enough to probe the local structure of water molecules in the liquid. What some groups began to find, still genuinely controversial, was that the single continuous network model might be incomplete. Liquid water might exist in two distinct structural forms, or fluctuate between two distinct local configurations. One form more tetrahedral, more ice-like in its local arrangement, the other more disordered, with weaker hydrogen bonds and higher density. The two forms interconvert constantly, but the balance between them changes with temperature and pressure in ways that might explain some of water's anomalies more accurately than the single network model does. This two-state model has supporters and vigorous opponents. Groups using different spectroscopic techniques get results that seem to support or contradict it, depending on how the experiments are designed.
In 2017, researchers at Stockholm University published results from X-ray laser experiments, suggesting that water at room temperature contains two distinct structural arrangements coexisting simultaneously. Others disputed the interpretation. The argument continues. What's striking about this debate is its location. Not at the frontier of exotic physics, not in the study of some rare material synthesized in a particle accelerator, but in the chemistry of the liquid making up roughly 60% of the human body. The structure of water inside you, surrounding every protein, making every biochemical reaction possible, is still not definitively described at the nanoscale. There's something almost vertiginous about that. We've sent spacecraft to the edges of the solar system. We've sequenced genomes. We've detected gravitational waves from collisions a billion light-years away.
And the liquid in the glass on your desk, its molecular arrangement, is still a matter of active scientific debate. A chemist who has spent 40 years studying water told a conference audience a few years ago that the more he learned, the less confident he felt in any simple picture of it. That isn't a confession of failure. It's what genuine progress sounds like when the subject is deep enough to resist easy description. Now sit with all of this for a moment. There's water near you right now, room temperature, unremarkable, sitting quietly. At its surface, molecules are escaping into the air, invisibly, carrying heat away, cooling the liquid by fractions of a degree. Below the surface, the hydrogen bond network is reforming and breaking at a rate too fast for any instrument to photograph in real time. Its local structure is something the best spectroscopy in the world is still trying to characterize. It looks perfectly still. In your body, the water is not still at all. It wraps around proteins, holds their folds in place, dissolves ions, and carries them through membranes, participates in the chemistry keeping your neurons firing and your muscles contracting and your cells dividing. Not a passive background, integral to the mechanism of you in a way that biology is still learning to describe precisely. Under the ice of Europa, tens of kilometers of fractured ice above a dark ocean that may be older than multicellular life on Earth, water sits in cold and pressure and darkness. Whether anything lives in that ocean, we don't know. The Europa Clipper is on its way to find out. But the water is there. It is there because ice floats, because water stays liquid under pressure, because the anomalies stack on top of each other in exactly the way that creates habitable conditions, even in the outer solar system, even without sunlight, even in a place that looks, from the outside, like nothing could possibly survive. Lawrence Henderson argued in 1913 that this was not coincidence. He didn't know about Europa. He didn't know about hydrothermal vents, or extremophiles, or RNA world hypothesis, or two-state models of water structure. He just looked at the list of what life needs and the list of what water provides and found them identical, item by item, anomaly by anomaly. He asked the question that still doesn't have a definitive answer. Is it possible that a molecule this precisely suited to life arrived at those properties by chance alone?
Physics doesn't answer questions like that. Chemistry doesn't either. They describe what is, with extraordinary precision, and leave the interpretation to whoever is paying attention. One molecule, 3 atoms, 1 bent angle. Bonds that break 1 trillion times per second. And from that, every ocean, every river, every cell, every thought, every living thing that has ever existed on this planet, and possibly on others. The water near you isn't waiting for you to make up your mind about what it is.
It's doing what it has always done. Breaking rules that needed breaking. Sustaining what needed sustaining. Hiding something about its own deepest structure that centuries of study haven't fully uncovered. We keep looking. We keep finding new rooms. If this video made water feel even slightly less ordinary to you, subscribe, leave a comment, tell me which anomaly hit hardest. This channel exists for exactly these moments, when something you've held in your hand your entire life turns out to be one of the strangest things in the universe.